Regulation of gene expression or regulation of protein synthesis in prokaryotes. In this chapter, we are going to learn regulation of gene activity, gene regulation in prokaryotes, coordinated regulation, strategies of gene regulation at transcription level, induction and repression, lactose system and lac operon system, allosteric regulation of lac repressor, role of cyclic EMP in catabolic repression, tryptophan operon system, attenuation or regulation after initiation of transcription, autoregulation, feedback inhibition, some topics related to gene regulation at translation level, gene regulation and bacteriophages, regulation of lytic pathway, regulation of lysogenic pathway, gene regulation in eukaryotes, genomic level at transcription level, control of gene expression at the post-transcriptional level. That means processing of RNA primary transcript level. Then control on transport of mRNA, control on transcription and translation of mRNA, control at mRNA degradation level, control at protein processing level, control at protein degradation level, evidence of regulation of gene action in eukaryotes. Regulation of gene activity. Inside every cell, DNA has the coded genetic information for all the proteins which may be required at different times during cell cycle. In spite of enormous genetic capacity, not all the genes function all the time or at any given time. As a matter of fact, out of 4,000 genes of bacterial genome and 1 lakh genes of human genome, only a fraction is expressed at a given time. Moreover, products of certain genes are needed only in certain types of cells. It means genes are selectively activated on special occasions in response to some prevailing environmental conditions and for a limited time on demand. In all the cell types, some enzymes are synthesized continuously all the time. Then it means the transcription of mRNA for much enzymes from constitutive genes occurs constantly. For this enzyme, for other enzymes, mRNA transcription is initiated only on demand and it is switched off when need is fulfilled. The control on gene expression or on protein synthesis is called gene regulation. The most efficient and economic way of controlling gene expression is the varying efficiency of promoter recognition by RNA polymerase and initiation of translation. But this gene control can influence the efficiency of different genes of the genome. Different mechanism will be needed to have products of those genes that are needed only occasionally and for those genes whose products are needed in significant amounts only under specific circumstances. This regulation of gene activity is associated with controlling quantitative differences in the amounts of proteins or enzymes of different genes, the amount of protein enzyme produced by a gene at different times or in different tissues. Exhaustive investigations have established that regulation of gene activity can occur at any one of the following levels: transcription level, at the time of synthesis of primary RNA transcript from the gene; then processing level, post-transcriptional level, at the time of post-transcriptional processing of primary RNA transcript into mRNA; translation level, at time of protein synthesis; then post-translation level, at the time of post-translational modification of polypeptide chain or chains into a functional protein. Then control of gene expression at the level of transcription initiation is the most economical way of control because this limits the number of mRNA molecules synthesized. The gene regulatory mechanism of prokaryotes and eukaryotes are slightly different and are discussed separately. Gene regulation in prokaryotes. In prokaryotes, the gene regulatory systems are adapted to provide maximum growth rate in a particular environment, and gene expression is controlled primarily at transcription level with some control at the translational level. Transcriptional control is accompanied by using on and off mechanism. It means a particular gene is switched on and synthesis of its mRNA is induced only when the gene product is needed. When the gene product is not needed, the gene is switched off and synthesis of its mRNA stops. The post-transcriptional control of translation level, is achieved by rapid degradation of mRNA molecules involved in this synthesis and modification of nascent polypeptide chain or chains into functional proteins. So coordinated gene regulation in bacterial systems, and several enzymes act in sequence in a single metabolic pathway. Usually either all or none of these enzymes are produced. This phenomenon is called coordination regulation. It results from control on the synthesis of a single polycystonic mRNA molecule that encodes all of the gene products. This type of regulation is not found in eukaryotes because eukaryotic mRNA is monocystronic. Then strategies of gene regulation. The bacterial cell protein synthesis of gene expression is mostly controlled at a transcription level. This regulation may be negative or positive. Negative regulation. In negative regulation, an inhibitor or repressor is present in the cell. The binding of inhibitor prevents or suppresses transcription of relevant gene. A slight molecule or inducer is needed to react with, and a signal molecule or inducer is needed to react with and remove the inhibitor so as to allow the initiation of transcription. The negative regulation of gene expression is also called repression. Then positive regulation. In this system, an effector or inducer molecule activates the promoter which promotes transcription of mRNA. There is no inhibitor-inducer interaction. The positive regulation of gene action is also called induction, and the substances that induce gene action is called inducer. Inducer may be a protein, a small molecule, or a molecular complex. Positive regulation can be achieved in any one of the two ways. Transcription will continue until a molecular signal binds to the activator molecule and cause it to dissociate from DNA. The activator molecular signal complex binds to promoter. If the signal molecule dissociates, the activator folds of DNA, stopping the transcription. Negative and positive regulation are not mutually exclusive. Some systems are both positively and negatively regulated. This utilizes two different regulators to respond to different conditions in the cell. A catabolic system can be regulated positively and negatively. For catabolic pathway, effectors are substrates which function as inducers. For anabolic pathways, effectors are usually the end products which function as repressors. Negative regulation, gene inhibitor, gene not expressed, but it removes inhibitor. What is that? Inducer. Inducer removes inhibitor, then inactive inhibitor will be there, so gene got expressed. And positive regulator, effector molecule bind to RNA, then that process will be happening. So the negative and positive regulation of mRNA transcription. A negative regulation inhibitor is attached to DNA and does not allow it to transcribe mRNA. Inducer binds to inhibitor and removes it from the DNA to allow mRNA transcription. And mechanism of gene regulation at a transcription level. Induction and repression. A set of genes will be switched on when there is necessity to metabolize a substrate. When these genes are switched on, enzymes are produced. This phenomenon is called induction. Similarly, the metabolite needed by bacterium is provided in excess in medium. Bacterium stops its synthesis and the genes associated with its metabolism is turned off. This is called feedback repression. Then induction and inducible system or inducible operon. In genetic induction, the gene or genes are induced or switched on to produce mRNA needed for the synthesis of required enzyme or enzymes. The substrate which induces the gene for protein synthesis or enzyme production is known as inducer. The phenomenon of induction can be demonstrated by growing E. coli in different nutritional media as follows. When Escherichia coli are grown in culture medium containing glycerol, they produce all those enzymes which are needed for the breakdown of glycerol. The synthesis of all other enzymes are maintained at the minimum level. When these E. coli are grown in a medium containing lactose, the synthesis of enzymes beta-galactosidase increase manifold. This enzyme hydrolyzes lactose into glucose and galactose. The synthesis of other genes is reduced considerably. When Escherichia coli are grown on glucose medium, they contain just traces of beta-galactosidase, an enzyme that hydrolyzes lactose into glucose and galactose. If this bacteria are transferred into a medium containing lactose, the concentration of this enzyme increases manifold that enables them to metabolize lactose. Here, substrate lactose has acted as inducer, activating the specific gene to synthesize the enzyme in pastin. On the above experiments, substrate glycerol and lactose act as inducers for the synthesis of required enzymes. The enzymes whose synthesis can be induced by adding substrate is called inducible enzymes. The gene complex responsible for the synthesis of inducible enzymes is called inducible system. Inducible system. Analysis of total mRNA present in the cells before and after addition of lactose shows that no lac mRNA is present before lactose is added to the medium. Addition of lactose triggers synthesis of lac mRNA. Here lactose is an inducer and the genes associated with the enzymes of lactose metabolism system form lac inducible system. Repression or a repressible system. In repression, the activity of genes or gene is suppressed and synthesis of specific protein is stopped or reduced. The substance which stops or suppresses protein synthesis is known as repressor. For example, in a minimal culture medium, E. coli cells can synthesize all the enzymes needed for the synthesis of different amino acids, but when a particular amino acid is added in the culture medium, the enzymes needed for its synthesis is not produced. When histidine is added in the bacterial culture medium, the production of histidine synthesizing enzyme is stopped. The enzyme whose synthesis is checked by the addition of end product is known as repressible enzymes. The genes associated with these enzymes form repressible system, and the substance that represses the synthesis of repressor enzymes is called repressor. As a matter of fact, some enzymes are normally present in the cell but ceases to be synthesized when high concentrations of their end products are present. These end products are called the co-repressors. The regulator gene produces a substance called aporepressor. The aporepressor unites with co-repressor to form the functional repressor molecule. This repressor molecule inhibits transcription of mRNA from all these structural genes which are responsible for the synthesis of enzymes of synthetic pathway. Most repressible enzyme systems occur in metabolic pathway. Difference between induction and repression. First, we are going to learn about induction. Induction involves switching on the specific gene to provide RNA or the synthesis of enzyme required. It is responsible for starting transcription and translation. It needs an inducer. When a new metabolite enters the system, it needs some new enzymes for its metabolism. Therefore, its concerned genes are switched on. Inducer does not permit the repressor to bind with operator gene. Inducer is associated with catabolic pathway. But repressor and repression is switched off. Repression is switching off the gene or operon which was actively producing an enzyme that is no more required by the cell. It stops transcription and consequently the translation. It needs a suppressor which acts as a feedback inhibitor. When end products of a metabolic reaction accumulate in the system, further reaction is not needed, and so also the associated enzymes. Hence, concerned genes are turned off. So, repressor is helped by a co-repressor combined with the operator gene. Repression acts in an anabolic pathway. The lactose system in E. coli. Jacob and Monod in 1961, while studying catabolism of lactose in E. coli, suggested that the action of most genes is regulated at the transcription level by induction and repression phenomena. Three enzymes, namely beta-galactosidase, galactose permease, and thiogalactosidase transacetylase, are needed for lactose catabolism. The genes or cisterns, DNA segments of these three enzymes are represented by LAC-Z, LAC-Y, and LAC-A. These are called structural genes and are located in a linear sequence in the bacterial chromosome. The action of these genes is controlled in a controlled fashion by regulatory genes. Lac operon system. Francis Jacob and Jacques Monod explained regulation of the activity of the SLAC genes by operon model. The operon model for lactose catabolism is called lac operon. It consists of many kinds of genes like structural gene. These genes are segments of DNA that are associated with the synthesis of these enzymes that are needed for catabolism of lactose. Structural genes. The lac operon, there are three structural genes which are the LAC-Z gene for beta-galactosidase enzyme, LAC-Y gene, LAC-Y gene for galactose permease enzyme, the LAC-A gene for thiogalactoside transacetylase. Thiogalactoside transacetylase. These genes produce a single polycistronic mRNA molecule. Francis Jacob, in collaboration with Monod, presented operon concept and concept of messenger RNA in 1961 and shared the 1965 Nobel Prize along with another scientist. So Francis Jacob and Jacques Monod served heroically during World War II. Jacob, as a member of the French forces, injured seriously in Normandy in August 1944, and Monod as the leader of the Paris Resistance Force. Monod had already obtained his PhD, but Jacob had to wait until 1947 to gain his MD from the University of Paris. Both spent the major part of their career at the Pasteur Institute, the famous Paris Research Center set up around Louis Pasteur in the late 19th century and still one of the most influenced European laboratories. So Francis Jacob contributed to some of the early work around bacteriophage with Andre Loaf and Elie Woldman prior to his collaboration with Monod that led to the operon theory and the concept of messenger RNA in 1961. The two shared the 1965 Nobel Prize with Loaf, both authored a number of good books. Then Control in the sense control genes, control genes or regulatory genes. These genes control the activity of structural genes and lie immediately adjacent to the structural genes. There are three control genes, namely operon gene. It lies just before upstream of first structural gene and overlaps the promoter sequence. It controls transcription of mRNA from structural genes. It is under the control of repressor protein produced by the regulator on repressor gene. Once again, operator gene, control gene on regulatory genes. Control genes or regulatory genes. These genes control the activity of structural genes and lie immediately adjacent to the structural gene. There are three controlled genes, namely operator gene. It lies just before upstream of first structural gene and overlap the promoter sequence. It controls transcription of mRNA from structural genes. It is under the control of repressor molecule produced by the regulator on repressor gene. Promoter gene. It lies immediately adjacent to the operator gene. RNA polymerase binds to this site. Transcription of an operon begins at promoter, a site where RNA polymerase binds for the transcription of mRNA from structural genes. It controls rate of mRNA synthesis. And repressor or regulator gene. It lies outside the operon and produces a repressor substance. This repressor binds to operator gene and suppresses the transcription of structural genes. Then functions of lac operon. Beckwith in 1967, Epstein and Beckwith in 1968. Repressor or regulator gene L. It lies outside the operon and produces a repressor substance. This repressor binds to operator gene and suppresses the transcription of structural gene, which are the three types of control genes or regulatory genes: operator gene, promoter gene, and repressor or regulator gene. In the absence of an inducer lactose, the regulation gene L produces a repressor protein R, which binds strongly to the operator site and prevents its transcription. As a result, the structural genes do not synthesize mRNA and proteins are not formed. When an inducer lactose is introduced in the medium, it enters the cell and gets modified in such a way that it binds to the repressor. The repressor now fails to bind to the operator. The operator is therefore free and induces the RNA polymerase to bind to the initiation site on promoter and to transcribe the cistrons into a polycistronic lac mRNA. This codes for the three enzymes necessary for lactose catabolism. Definition of operon. An operon is a group of closely linked structural genes or cistrons and the associated control genes, operator and promoter genes, which regulate the genetically controlled metabolic activity. And structure of operon. The genes composing operon are classified into two categories: structural genes. The structure genes are those segments of DNA which carry codes for the synthesis of proteins. These genes determine the primary structure of polypeptide chain by controlling the sequence of amino acids during protein synthesis. Then control genes. This control the activity of protein genes either by induction or by suppression. These genes are regulator gene. The regulator gene L produces some specific enzymes which acts as a repressor substance. In case of lac operon, the regulator gene codes for a protein which strongly binds to the operator gene and suppresses its activity. Then promoter gene. The promoter gene is the DNA segment at which RNA polymerase binds and initiates to transcription of the structural gene. Operator gene. The operator gene is a segment of DNA which exercises a control over transcription. The repressor substance produced by the regulator gene binds with the operator gene. It lies close to the structural gene. RNA polymerase unable to bind to promoter. Repressor bound to operator. Lac Z gene, lac Y gene, lac A gene, no mRNA, active form of repressor, like that. Function of lac operon in Escherichia coli. Lactose absent, repressor active and bound to operator, operon repressed, transcription blocked and no mRNA synthesized. Lactose present, repressor bound to lactose and inactivated. The repressor no longer binds the operator. Operon repressed. Transcription begins and mRNA synthesized. repressor proteins are allosteric. Those exist in two conformational forms. In one form, the protein is active, and in the other form, it is inactive. When effector molecule binds to the protein molecule undergoes conformational change and becomes inactive. But change is irreversible. So, as soon as effector moves away, protein becomes active again by returning to the original form. Role of cyclic AMP in catabolite repression, positive control. Small molecules of cyclic AMP, CAMP, are distributed in bacteria and multicellular eukaryotes. Cyclic AMP is synthesized enzymatically by adenylyl cyclase. Its concentration is regulated indirectly by glucose metabolism. When bacteria are growing in culture medium containing glucose, the CAMP concentration of the cells is quite low. In a medium containing glycerol or any other carbon source that cannot enter the biochemical pathway used to metabolize glucose, the concentration of CAMP become high. It regulates the activity of lac operon as well as for some other operons. E. coli contains a protein called catabolite activator protein or CAP. It is encoded by a gene CRP. Catabolite activator protein CRP coded by CRP. Mutants of either CRP or adenylyl cyclase gene are unable to synthesize lac mRNA. It means both CAP and CAMP are needed for lac mRNA synthesis. CAP and CAMP bind together, forming CAMP-CAP unit. The complex unit regulates LAC system. It is a positive regulation. It functions independent of the LAC repressor system. In the absence of CAMP-CAP complex, RNA polymerase binds weakly to promote the weak binding fails to initiate transcription. So CAMP-CRP complex binds to a base sequence in RNA in the pioneer region of the initiated transcription. Then tryptophan operon, a repressible operon system. Tryptophan operon in E. coli is responsible for controlling synthesis of amino acids. Tryptophan, its synthesis is carried out by 5 enzymes. The enzymes including these enzymes are located adjacent to one another in suppression and is termed TRPE, TRPD, and TRPC and TRPE. TRPA. Then gene TRPE is the first one to be translated. Genes promoter, operator, and two regions called leader and attenuator are located close to gene TRPE. The repressor gene TRPR is present far from the gene cluster. Tryptophan operon is switched on for normal transcription when tryptophan amino acid is not accumulated in the medium in short supply. This operon is switched off if only when tryptophan accumulates in the medium. The functioning of tryptophan operon in E. coli: when tryptophan is present, it binds to repressor. The activated repressor binds to operator and operon is repressed. When tryptophan is absent, the repressor does not bind to operator and operon actively transcribes mRNA for enzymes. The regulatory protein of TRP operon is produced from the TRP gene. So mutations either in this gene or in the operator gene cause constitutive initiation of transcription of TRP mRNA. This protein synthesized from mutant gene is called aporepressor. It does not bind to the operator unless tryptophan is present. The aporepressor and tryptophan molecule join to form active TRP repressor which binds to the operator to switch it off. So aporepressor, no tryptophan, active repressor, active operator, transcription, aporepressor, tryptophan, active repressor, operation, switched on. Aporepressor, no tryptophan, active operator, transcription. But when aporepressor plus tryptophan, active repressor, not operator, active repressor, operator switched off, no transcription. And attenuation, regulation after the initiation of transcription. In most cases, regulatory mechanisms control the initiation of transcription. In prokaryotes, some regulatory mechanism exercises control following the initiation. Charles Janowski and his colleagues found that TRP operon has a regulatory site between promoter and operator and has structural gene TRP. The site of DNA is called a leader sequence. It is non-coding DNA segment which is transcribed into leader mRNA at the 5' end of mRNA. This TRP leader mRNA is 162 nucleotides long. It is sensitive to tryptophan level and exercise an additional control on the transcription of polycystonic TRP mRNA. The effect of this controlled element, leader sequence, is called attenuation because it reduces an attenuated synthesis of mRNA. Structure of leader mRNA. The 162 to base long leader mRNA as following segments. It has an initiator codon AUG at 5' end and a stop codon UGA at the same reading frame. This region encodes a polypeptide consisting of 14 amino acids. It is called leader polypeptide. The leader polypeptide mRNA contains two adjacent tryptophan codons, position 10 and 11. It means the 10th and 11th amino acids in leader polypeptide and tryptophan. Leader mRNA has four distinct regions denoted as 1, 2, 3 and 4. These regions can base pair with each other in different ways, forming several hairpin structures. For example, in a stable form, the free leader mRNA shows base pairing between 1, 2 and between 3 and 4 regions. In low concentration of tryptophan, ribosome is stalled in region 1 so that region 2 and 3 base pair together. Transcription of region 4 and subsequent structure genes are completed. In high concentration of tryptophan in the medium, the 3 and 4 regions get base paired and synthesis of protein is interrupted by on reaching the second region. After region 4, it's present a string of A to U trying just prior to the structure gene TRPE. A part of leader mRNA comprising region 3 and 4 and string of A to U is called attenuator. When base pairing between 3 and 4 regions create a hairpin, the attenuator acts as a transcription terminator signal and RNA polymerase and growing RNA chain detaches from DNA. The mechanism of attenuation. During translation, a ribosome attaches to the first bone binding site, the initiation codon on TRP mRNA. Transcription of mRNA and its translation follow closely. When tryptophan level in the medium is low, the transcription of tryptophanyl tRNA molecule carrying tryptophan is also low. When ribosome arrives at the regulation of DNA gene expression by tryptophan codons in leader sequence, it waits for the arrival of tryptophan tRNA and blocks region 1. As a result, the hairpin structure is formed by pairing between region 2 and 3. Thus, region 3 is unable to pair and form hairpin with region 4. The termination of hairpin loop is not formed, and transcription of structural genes of tryptophan operon continues to produce polycystonic TRP mRNA. When a tryptophan level in the medium is high, the tryptophanyl tRNA concentration is also high. The ribosome moving from the initiation codon passing over tryptophan codons and stops at stop codon. Thus, it blocks region 2 of TRP mRNA. This permits pairing of segments 3 and 4 and 3-4 hairpin loop is formed. This, along with poly U, causes the termination of mRNA and structural genes are not transcribed, stopping the tryptophan synthesis. A regulatory mechanism of this type cannot exist in eukaryotes because transcription and translation cannot be coupled. In eukaryote, transcription occurs in the nucleus and translation in the cytoplasm. Then autoregulation. Synthesis of monocytonic mRNA can be autoregulated by gene product acting as a repressor. In an autoregulated system, when concentration of gene product exceeds than needed by the cell, the gene product molecule binds to the operator inhibiting transcription. When concentration of gene product falls below the required level, there is either due to use or increased need, the molecule bound to the operator leaves the site and operator and the promoter are free, leading to the onset of transcription. So synthesis of most repressor proteins and many enzymes needed at all times are autoregulated. Then feedback inhibition and end production inhibition. In case of end product of a biosynthetic active pathway is not utilized, it accumulates inside the cell and stops the synthesis of that substance. This is known as feedback inhibition of the gene action. The inhibition is caused by the binding of the accumulated end product to the enzyme. For example, isoleucine synthesis in E. coli can be inhibited in some isoleucine is called to the medium. By the binding of isoleucine to the first enzyme, the enzyme is inactivated. This phenomenon of inhibition of enzyme level is known as allosteric interaction. Thus, in feedback end product inhibition, the accumulated end product of a biosynthetic pathway combines loosely with the first enzyme in the pathway. This complex does not bind to the operator or promoter site, but the tertiary structure of enzyme is modified, which makes the enzyme inactive. This allosteric transition of enzyme molecule blocks its enzyme activity and prevents overproduction of end product and also the intermediate metabolites. Then in the end product inhibition, the expression of gene is regulated after proteins have been synthesized. This is a post-translational control of gene action. Gene regulation at translation level. Translational regulation of protein synthesis refers to the control of members of a copies of particular protein translation from a polycystonic mRNA. It varies from gene to gene, and there is a gradient translation increasing from 5' terminus to 3' terminus of mRNA. This depends on varying efficiency of the initiation of translation, different spacing between chain termination codon and next initiation codon, different sensitivity of various regions of mRNA to degradation, then inhibition of particular gene by its gene product, feedback inhibition, synthesis of ribosomes. Gene regulation in bacteriophages. Gene expression in bacteriophages is also regulated through operons. There are promoters and repressor gene which regulate the activity of structural gene. For example, and the phage is going to follow lytic or lysogenic pathway is determined by genetic switch involving repressor C and CRO gene. Switching of a CRO gene initiates lytic pathway, while turning off repressor gene CL leads to lysogenic pathway. Then regulation of lytic pathway when repressor gene is inactive. Two promoter genes PL promoter left and PR promoter right are transcribed. Gene PR synthesized CRO protein switches in the lytic pathway. When PL transcribes protein N, it allows RNA synthesis to proceed over certain transcription terminators. This process is called anti-termination, and gene PL is anti-terminator gene. The products of both these genes turn on various genes of lytic pathway. They promote synthesis of coat protein, replication of viral DNA, assembly of new phage particles, and lysis of bacterial cell. Then regulation of lysogenic pathway. Lysogenic pathway is initiated by protein produced by C11 and C3 genes. This activates transcription of CI gene, which produce lambda repressor protein. It binds to two operators OL and OR also, and it inactivates PL and PR. As a result, transcription of CRO and N gene is blocked. Thus, in bacteriophages, genes related to lytic or lysogenic pathways are organized into operons. Like bacterial operons, these operons are controlled through the interaction regulatory protein with operons, and these operons lie adjacent to clusters of structural genes.